think we are good to get started. I would like to welcome everybody again to the Morphology That Matters webinar. This is ranging from cell quality to drug response. We're going to be listening to two leading researchers demonstrate how they are using REMI to unlock morphological insights into their research, which is pretty amazing. So I'm very excited for today. Thank you so much for joining. I will start by jumping into a brief overview of the REMI platform. So I am Megan. I'm a field application scientist at DeepCell. I've been working for DeepCell for around 3 years. I've been working very closely with Dr. Caroline Heckman and Dr. Jean Rivera over the past couple of months, and I just am so excited to be able to watch them, share their research and share how they're implementing REMI into their research projects lately. They're very impressive. I can't wait for you all to hear what they're doing. I'm just going to begin by explaining a little bit about the REMI platform for anybody that is new, and then we will just jump right into their presentations. Thank you so much for joining. All right. So morphology is the bedrock of cell biology. Morphology is the study of form and structure. Cellular morphology forms the foundation of cell biology, as the structural organization of cells underlies their functional capabilities, whether that's cellular division, motility, or a cell's ability to launch an immune defense. There are 100 trillion cells in the human body, consisting of 200 unique cell types and counting, each diverse as it relates to their state and function. The ability to understand heterogeneity lying within those 100 trillion cells is fundamental to understanding mechanisms of disease and other conditions plaguing society today, and enables us to progress towards better treatment options. Morphological characteristics provide a window into a cell's physiological state or pathological condition, both of which we will actually explore today, making morphology a cornerstone in experimental and clinical contexts. While molecular diagnostics and multi-omics technologies are advancing rapidly, morphological assessment remains indispensable for disease diagnosis, monitoring progression, and guiding therapy. Current approaches to analyzing cellular morphology are often limited in scale and precision, rely on labels, and typically cannot efficiently sort out cells or preserve their viability for downstream functional assays. Developing higher throughput, higher dimensional methods is therefore essential to fully link cellular phenotype with function and unlock deeper insights into biology and disease. Multi-omics approaches are rooted in the central dogma and expand it into a multidimensional framework that captures the dynamic, interconnected nature of cellular systems. Including each ohm reveals a unique layer of biological complexity, but none alone captures the whole picture. Turning cell morphology into a quantitative high-dimensional ohm allows us to understand the dynamic state of a cell in space and time, while also maintaining their naive, unperturbed conditions. Assessing morphology in a label-free manner limits bias and allows the culmination of the genome, transcriptome, and proteome of a single cell to drive the biological insights rather than relying solely on markers sitting on the surface of a cell. Now here's where the REMI comes into play. The REMI takes high-resolution bright field images of single cells in their most natural state and analyzes them using AI to enable quantitative morphological profiling. On top of that profiling, the REMI takes morphological profiling a step further by enabling researchers to gently sort morphotypes of interest, keeping their cells in their most unperturbed state for downstream assays. I really like this slide because it shows the high-resolution images that our system captures. Also shows the instrument itself, but there's just such a wide range of diversity in the cell types that we image. Honestly, morphology applies to so many different applications in biology, whether this is drug resistance, drug discovery, oncology, um, immunohistology. There's just so many different areas where morphology applies in order to connect the different omics. And using these high-dimensional, high-resolution images is just the first step into unlocking those insights. It's pretty interesting where working with customers, we have yet to find an application where morphology doesn't at least reveal some sort of insight or at least a new perspective onto traditional methods of analyzing application areas. Going into the REMI instrument itself, The REMI sits on a benchtop and can be used with nearly any cell type or experimental design. With the input of the system being a single cell suspension, the only sample processing required is a simple buffer exchange, which makes my life easier. Using microfluidics, the cells then flow through the instrument at a high speed, get their picture taken in high resolution, and are characterized in real time using 115 dimensions by our foundation model. Led by user-generated classifiers, cells can then be sorted gently 6 ways and collected after a run for downstream processing. So you can see the instrument to the right here. You see that the workflow is quite simple. The input is the sample tube on the right-hand side. We have a microfluidic chip, which is where the magic happens. The cells simply flow using microfluidics through our chip, get their image taken, and whether you are imaging or you are sorting, we provide flexible workflows for researchers to participate in either. If you are sorting, you would simply then just collect your cells from 6 wells on our microfluidic chip with a P20 pipette. Pretty amazing. Now going into the human foundation model. The human foundation model is the bread and butter of how we integrate AI into cellular morphological analysis. The human foundation model acts as a feature extractor, with the input being a high-resolution, bright-field image of a cell, and each image passes through both a deep learning encoder, which generates 64 deep learning embeddings that we use to plot our visualization projections, and it also goes through a computer vision encoder, which enables us to understand morphological readouts in a human interpretable manner. These are things like cell size, cell shape, pixel intensity, texture, granularity, all combined together. So we look at a cell from 115 different perspectives for each individual cell image. By combining deep learning and morphometric features, the Human Foundation Model, or otherwise known as HFM, provides both accuracy and interpretability. The data that comes out of HFM can be depicted in morphology UMaps, very similar to how we typically visualize single-cell RNA sequencing data. Cells that have similar morphology cluster together, which allows Leiden clusters to guide our understanding of morphological heterogeneity of a sample without the need for surface markers or staining. We can also use morphology density UMaps, which we see below, to understand differences between certain samples, or honestly, any sample that you run on our system. Comparing whether this is tracking a differentiation progress, comparing drug treatment time points, or simply comparing a healthy sample to a diseased one, the morphology density UMaps are very powerful visualization tools that I personally use every single day in my analysis. These projection methods and analysis tools make it very easy for researchers to identify morphotypes of interest in an unbiased manner. Sorting out these morphotypes is as easy as making a simple lasso selection and clicking a button. This is what our sorting looks like in real time. We have a very simple, very gentle method of sorting, which essentially keeps these cells viable for downstream processing and any downstream functional assays you would like to do. Oftentimes people sequence after sorting these cells, but we've had people, experiment quite nicely with different functional assays after the fact. The world is your oyster when you're looking at how to use these viable, unperturbed cells. Now, I would like to pass the microphone over to two leading researchers who are using this next-gen technology to push the boundaries of what's possible. I'm so happy to introduce Dr. Caroline Heckman, Research Director at the Institute for Molecular Medicine, Finland, and Dr. Jean Rivera, Cell Metrology Team Leader and Science Lead at the UK National Measurement Laboratory. Thank you so much, and Caroline, please take it away.
All right, thank you, Megan, and thank you for the Deep Cell team for the nice invitation to speak today at this webinar. And just get my slides set up. Hopefully everybody can see the slides okay. And anyway, I'll tell you a little bit about what we have been doing here at my Institute for Molecular Medicine Finland, which is part of the University of Helsinki, and the research that we have been doing, especially with acute myeloid leukemia, and applying the deep cell technology to understanding drug response in this disease. So I'm going to give you a bit of an overview of the research that we have been doing here for the past 15 years in terms of functional assessment of blood cancers and response to specific drugs and trying to identify the right treatment for patients and also the different platforms that we have applied and then how those platforms have been applied to guide the treatment decision in the clinic. But then I also want to show you what we recently were able to do using the DeepSoft technology with the REM-I instrument and also the human foundation model that Megan just described, and which is, I think, pushing our research into a new dimension. And it's really exciting. I look forward to continue down that path. All right, so over, I think, 15 years ago, we started here at our institute a program which we called the Individualized Systems Medicine Program for Hematology. And the idea for this program was to set up a biobanking infrastructure for patients with blood cancers. Basically, their samples would be collected at different time points of their disease, and they would be stored so that they could be used for different research assays and basically viably cryopreserved. Some of these assays included sort of standard next generation profiling analysis, including whole exome sequencing and bulk RNA sequencing, but also high throughput drug sensitivity testing. And the idea was to integrate the data from these different platforms so we would get a much better idea of the disease biology, get a better idea of response indicators, but then also be able to feed that data back into the clinic in case it would be useful for patients who are no longer responding to the standard of care. So that type of data would then be discussed through a tumor board. So just to mention, I think it was really important. One of the foundations for our research was to establish this national biobank. So Finland, there are only 5.5 million people, and these blood cancers, the number of patients per year is relatively small. So for a disease like AML, we might only see 200 to 300 patients with newly diagnosed disease, and it was important that to have a standardized repository and also a clinical registry that could match those samples to the clinical data. And then at the same time, we were setting up our next generation sequencing assays at our institute and our platforms for looking at somatic mutations, for example, in cancer, but then also more recently, of course, single cell technologies as well. But then we've also had very nice high throughput capabilities, mainly where we've been able to apply patient cells to hundreds of different drugs and analyze them using different platforms. So when we first started this project, the techniques that were used were basic bulk viability assays. So this is sort of your typical MTT assay or cell titer glow assay, which is just giving you a readout of all the cells that are in your cell culture plate or in your well. And so you don't get a grasp of the heterogeneity of the patient samples or patient tumor, but at least you get sort of a general efficacy. So if most of the cells in the well are tumor cells, then the readout should basically give you an indication of how that drug's affecting the tumor cells. Over the course of a number of years, we progressed as high throughput flow cytometry technologies progressed as well. We adopted those, and since we were studying blood cancers, it made sense to start analyzing our patient cells with that type of technology, where we could then get a dose response curve, for example, for every cell population that we were interested in looking at in these mixed patient populations. And at the same time, we could also use high count imaging. This was particularly useful for cancers that were not necessarily blood cancers, but for groups that were looking at solid tumors. For example, we can't do as much multiplexing with this platform, but certainly can look at different cell populations within a mixed culture. But with all of these technologies, you're just getting sort of a basic readout of different cell types. You might get the bulk viability readout, but then with high throughput flow cytometry and also multiplexed imaging, you can look at different cell populations and get a sense of how the drugs are affecting those cell populations. So this was also based on being able to use hundreds of different drugs, either investigational or approved oncology drugs that could be tested in different concentrations. And all of these drugs, and currently we use about 525, are pre-plated on 384 well plates and stored until the patient sample arrives. And when they arrive, we can just add them to the plate, incubate the cells with the drugs, and then do the readout. So basically, it would be either the bulk viability, or we could do a flow or imaging-based readout. And then the computational scientists also at our institute developed ways for us to analyze this data more efficiently. So basically, we could create a score, a drug sensitivity score, that would allow us to differentiate the sensitivity of tumor cells versus healthy cells, and then prioritize those drugs that would be more effective on the tumor cells. So after doing this for many years, we published some results for relapse refractory AML and how drugs could be repurposed and utilized for patients who are no longer responding to the standard of care. So basically, when we were starting this project, there was only a two-drug chemotherapy combination that was being used for these patients, and then bone marrow transplant. But over the years, many new targeted drugs have been developed and approved for AML, but nevertheless, most patients who have relapse disease have very poor outcome and are very challenging to treat. So this was a group of patients that we focused for trying to find new treatment options. And just to give you an idea, this is almost 40 patients that were treated with drugs that were determined to be effective by our ex vivo assay. And many of these patients did quite well in terms of the initial response, and they were able to be bridged to a bone marrow transplant. But basically, if they had not received this option, their outcome would have been relatively poor, but we were able to see an overall response rate of 59% compared to perhaps 25%. So this was quite a nice accomplishment. But then another thing we wanted to know was also if our ex vivo assay would actually be predictive of patient response. So one of the new drugs and drug combinations that was approved for AML while we started this project was the BCL2 inhibitor venetoclax in combination with the hypomethylating agent azacitidine. So my colleague Mika Contro and also another colleague, Heikki Kusimäki, who are both former students from the group, they started a clinical study called the VENIC study. And the idea with this was it was a two-arm prospective study that was recruiting AML patients at diagnosis, also relapse refractory patients. and therapy related AMLs as well. And these patients, their cells would be screened by our ex vivo assay, and then they would be given the treatment, but the clinician, who is Mika, would not actually look at the ex vivo results. But most patients were given the two drug combination, and then later there was an ex vivo in vivo correlation. So for this, for the setup of the correlative studies, bone marrow samples were taken, and then the mononuclear cells were enriched, and the cells were either screened using a bulk assay, which was cell titer glow, or our flow-based assay. And then afterwards, the results were correlated to the outcome of the patients. At the same time, they wanted to test different cell culture medias, because we often see that the media type can affect the drug response as well. So the cells were tested in three types of media: just a basic RPMI media, and then a conditioned media. So we took conditioned media from the HS5 human bone marrow stromal cell line and mixed it with RPMI. And then another commercial option, which was from stem cell technology, which had a defined growth factor cytokine mix. So doing the analysis by flow-based analysis, and then also by bulk viability readout by cell titer glow, the group could see that for patients that had refractory disease or progressive disease, their ex vivo response was quite low compared to those patients who had a complete response. And just for your information, this DSS, a drug sensitivity score, the higher the DSS value, the better the sensitivity of the cells to the drugs. But basically, yeah, there was a significant difference between the ex vivo response with patients who had refractory disease and those patients who had a complete response. So interestingly, when we're comparing the different types of media, the conditioned media, which was sort of a in-house made media, performed the best, and compared to a standard RPMI mix or the stem cell technology mix. And this was also not only the case for venetoclax itself, but also with the combination of venetoclax and azacitidine. In addition, the flow cytometry assay or platform was a much better readout compared to the bulk viability readout. But overall, yeah, we could see that ex vivo sensitivity to venetoclax could predict treatment response, and so those patients who had a complete response, generally, there was very good sensitivity, and compared to those patients who had relapsed disease. So this was a nice validation, a clinical validation that the functional testing, or ex vivo drug sensitivity testing, could predict patient outcome. And also at the same time, we could see that if the ex vivo sensitivity was good, the overall survival of those patients was very good compared to those patients whose cells were ex vivo resistant. And this was the case for both previously untreated or newly diagnosed AML, as well as relapsed and refractory or secondary AML. So the next question then, based on this historical data, was when Deep Cell approached us, and the Deep Cell team approached us, was, well, if this was the case, that from our previous knowledge about ex vivo testing and prediction of patient response, well, we also wondered whether morphology can also distinguish between AML patients who might be sensitive or resistant to a specific treatment. And this was also based on a paper that was published several years ago when Heikki was a PhD student in my group. So what he showed was that with several targeted drugs, like the MEK inhibitor, trametinib, or JAK inhibitor, ruxolitinib, also venetoclax, we could clearly see that there is a difference in response between different cell subtypes within the same patient sample. So, for example, with the MEK inhibitor, what we could see is that immature blasts or hematopoietic stem and progenitor cell populations were tended to be more resistant to MEK inhibition compared to monocytic cell types, so a more mature cell type. And this was also the case for the JAK inhibitor, ruxolitinib. But if we looked at venetoclax, for example, the less mature cells tended to be more sensitive compared to more mature cell types. So this gave us the idea that actually we know that if there is an association between cell state or cell maturity and targeted drug treatment, we know that these cell states also have a different morphological type as well. So we hypothesized that maybe morphology would also be predictive of cell response to these targeted drugs. So this is the question that we asked when Deep Cell approached us and asked if we would be interested in testing the REM-I instrument. And so what we did was a team from the group, June, Ella, Danielle, and Mina, took the samples from the patients that were involved in that VENEX study, and then also a set of healthy bone marrow cells and screened them on the REM-I. So basically a set of 6 healthy donor samples, bone marrow cells from healthy donors, 5 samples that we knew were from patients who were venetoclax sensitive, and then also 5 samples from patients who were venetoclax resistant. And for all of these samples, 25 to 50,000 images were taken. And if we look at the analysis of all of the samples together, so all 6 healthy samples and all 10 AML samples, what we could see is a very nice distinction between the healthy samples and the AML samples. So basically these these density heat map, UMAPs are showing us that with the healthy bone marrow samples, you get actually a spread across the UMAP. But with the AML samples, it's much more concentrated. And when we think of AML samples, we know that usually for newly diagnosed AML, maybe the blast count so the tumor cell content is probably about 80%, often at diagnosis. So you're getting like 80% of cells that look very much alike. And this is very apparent in this type of analysis that we did on the REM-I instrument. So you see that this nice tight cluster of AML cells compared to the healthy bone marrow cells, which are much more diverse and then also representing the many different clusters that were detected using the Human Foundation model. And then if we look at each of those samples individually, you can see diversity, of course, among the different healthy donor samples, but sort of a similar widespread. While with the venetoclax sensitive AML cells, which are mainly the newly diagnosed, untreated samples, what we're seeing is really a nice tight cluster of cells in all of these 5 samples. Interestingly, for the venetoclax resistant AML samples, we're seeing not so much of except for maybe one case, we're seeing a bigger spread of cells, a little bit reminiscent also of the healthy donors, but also very distinct as well. And this was also the case with the blast cell content. So the blast cell content was actually higher with these venetoclax sensitive AMLs compared to the venetoclax resistant AMLs. We also took a look at the morphology changes that might be occurring during disease progression. So for a few patients, we had paired samples. So a sample taken at diagnosis before treatment, then also after treatment at relapse. And you could also see a nice shift in morphology as well, and also changes in the individual cell images. But if we look at the data that's coming off of the Human Foundation model and the differences that are detected there, actually it was the deep learning uh, features that were the most distinct, that made the distinction between the diagnostic sample and the relapse sample. So basically, we would probably not be able to see these differences really by eye, and this is also sort of apparent from these example images. But I think just with this one project, we really got a sense that morphology is definitely playing a role in perhaps patient response. We had already published the results showing that the differentiation status of AML is important for response to target drug treatments. But it seems like morphology is also playing a role. And this really makes sense because the different the cell differentiation states in AML, the cells do look different, and so they will have a different morphology. And this is definitely being picked up by the deep cell technology with REM-I and the Human Foundation model. So it was really nice to see this validation and the instrument being able to pick this up. And we're really looking forward to pushing this forward and investigating a bit further, how we can use morphology to predict response, not only to a drug like venetoclax, but to other drugs as well, and also other blood cancers. So it was really a nice opportunity, and I'm really grateful to the Deep Cell team for being able to allow us to use this technology, but then also my local team and my group. So June was really leading the way with our Venex sample analysis, and then supported also by Danielle, Amina, and Ella, and then also grateful to Mika and Heikki for providing these samples for our analysis, but then also Deep Cell for their help, and Megan especially, and providing the training, and then the help with also the analysis too. So thank you so much. I'll take your questions hopefully later.
Awesome. Thank you so much, Caroline. I love that photo. It's so fun to always get the photos with all the teams afterwards. It was such a pleasure to work with your team. Thank you so much for sharing your work with the larger group and with the larger audience. And I can't wait for what's to come. I do think we have a question. We can maybe answer like one or two now, and then we'll definitely be able to save time. I want to be conscious of time for Jean's presentation, so we can have a larger Q&A session at the end of the webinar. But there is one question. So someone asked, why did you prefer a more unbiased approach for your analysis? How did classic markers limit your research?
Yeah, well, that's an excellent question because for classical analysis like flow, we are limited to what we know. So the morphological analysis would probably provide us with a lot more ability to follow up with perhaps cell populations that are not detected by the classical flow markers. And so this is actually something that we're looking into right now. So we're looking, especially with the resistant samples, at clusters that are showing up in those samples that might be associated with resistant disease, and that we could follow up later with some additional analysis, for example, proteomics or single cell analysis. So it'll give us I think hopefully new ideas of drug resistance and perhaps ways to target those cells.
Awesome. Thank you so much. I do see another question, but I think we'll probably hold that one off until the end because it is 40 minutes past the hour. So thank you so much again, Dr. Heckman. I would love to pass the mic off to Dr. Rivera. Thank you so much.
Everyone, let me just share my screen. Hope everyone can see that okay.
Looks great.
Yeah, thank you. So yeah, what I wanted to do today is just kind of delve into some of the work that we've done with DeepCell in regards to redefining cell quality and standardizing morphological profiling as a critical quality attribute in ATMP production. So firstly, I'd like to thank everyone for joining the webinar and for DeepCell for allowing us to actually show some of our data, exciting data that we got from DeepCell demo here in Leeds. I'm Jan Rivera. I'm the science lead for cell biology at the National Measurement Laboratories. And I'll be specifically looking into CQAs in terms of the production of advanced therapy medicinal products, or ATMPs for short. So this work is part of our broader mission to improve measurement science in cell biology and also support innovation in biomanufacturing. So before we dive into the science, I just wanted to briefly introduce the National Metrology Laboratories at LGC. We're the UK's designated institute for chemical and biological measurements, and we play a key role in the UK national measurement system. So we ensure that the work is traceable within the chemical and biological measurements, such as nucleic acid quantification, nanoparticle concentration, chemical purities, proteins, and cell analysis. So we actually support industry, academia, and government by providing reference standards, reference methodologies, and also calibration services. Our headquarters is based in Guildford, but the sales team is in Leeds, where we had the opportunity to evaluate DeepCell. We also have strategic partnerships with other universities, such as University of Liverpool, Queen's, University of Belfast, and University of Strathclyde, all strategically positioned to drive regional innovation in clinical sciences, diagnostics, and also agriculture. So in terms of ATMP production, standards are essential to ensure product quality, safety, and efficacy. And these are shaped by regulatory frameworks like GMP and guidelines derived from MHRA in the UK, European Medicines Agency in Europe, and FDA in America. There's various international initiatives, such as the International Council for Harmonization guidelines that defines key concepts like quality target product profile, critical quality attributes, and critical process parameters. So for this specific talk, I'll be discussing cell critical quality attributes for cell therapies that are measurable characteristics. So this can either be physical, chemical, or biological, or microbiological, that can be quantified to and directly impact product performance. So what I mean about CQAs is a number of measurands, including identity, purity, potency, safety, quantity, stability, viability, and morphology. So all these attributes ensure the product is safe, effective, and consistent. But due to biological variation, defining and measuring these CQAs early in development is challenging, but also quite crucial, because failure to do so can lead to late-stage clinical trial failures, for example, or safety concerns post-market. In particular, I wanted to focus in on morphology, as this is the topic of discussion in this talk, that refers to size, shape, and structure of cells. I will also be delving into and mentioning aspects of viability and also quantity. So what I mean about cell morphology as a CQA is mainly morphological profiling and its emerging role in cell therapy quality control. So morphological profiling, as Megan mentioned, is an unbiased imaging-based approach that allows us to interrogate physical characteristics of cells. This can be shape, size, and texture to generate these unique phenotypic signatures. So these signatures can then be used to cluster cell populations and predict bioactivity in response to perturbations. So particularly valuable in ATMP production, where understanding these subtle changes in cell phenotype can actually be quite critical in ensuring product consistency and safety. And in the last few years, there's a lot of recent technological advances that have enabled broader use of label-free imaging modalities such as bright field and phase contrast. All these techniques allow us to observe the cells in their native state without the need for fluorescent markers or dyes, which can sometimes interfere with cell function or viability, for example. So one of the most exciting developments is the integration of image-based flow cytometry that combines bright field imaging with traditional flow cytometry. So this allows us to accurately identify different cell clusters and cell populations that enhances our ability to detect outliers or stressed cells. So for example, here on the top panel, you can see the differences in terms of accuracy in identifying single cell populations, as referred to as R1 and R2 here, using conventional gating strategies, as opposed to image-based single cell gating strategies. And you can clearly see here that the approach of image-based gating strategy can actually allow us to improve our accuracy in only isolating single cell populations or very specific clusters, if that's what we're interested in. So in particular, this field is now formally recognized through ISO standards. So there's a standards under biotechnology that specifically looks at cellular morphological analysis and highlights these requirements for cell morphometry to quantify cell morphological features. So this standardization is actually a key step in making morphological profiling a regulated and recognized quality attribute of cells. So why does this matter in terms of ATMPs, where morphological profiling can actually help elucidate mechanisms of actions, specifically in complex cell therapies, for example, and also guide future manufacturing strategies by identifying phenotypic markers that could be linked to potency or stability. It can also provide a more novel approach through identification of CQAs through omics approaches, as again, Megan mentioned previously. So in short, morphological profiling is not just a diagnostic tool, it's a strategic asset in biomanufacturing, as it helps us move beyond the traditional viability assays and towards a more real-time understanding of cell health and functioning. So what I wanted to show you here is the ability of DeepCell as a tool to be used as a quality assurance assessment of controls. So as I mentioned, cell morphology plays a critical role in controlling and understanding cellular health, behavior, and function. And it can actually vary based on cell type, growth phase, and environmental conditions. And changes within these morphology can indicate cell stress, contamination, or disease. And morphological shifts can actually reveal undesired changes in cell phenotype, which is crucial in long-term cultures, for example. So maintaining the healthy morphology is essential for maximizing yield and reproducibility in biomanufacturing and tissue engineering. So what we've initially seen in DeepCell is that it is able to actually identify these subtle morphological changes that I've mentioned could otherwise be missed. So for example, in this example here in MRC5s, samples, features like area, perimeter, and centroid axis reveal differences in cell cycle progression across the three different replicates of what we initially thought were healthy optimal samples within the optimal passages. So this suggests to us that the samples may actually be different in terms of cell cycle, which could be attributed to its different passages. And this shows us that actually being able to hone in and custom provide thresholds for what we are determining as the healthy or control sample could actually be critical in maintaining consistency in biomanufacturing and understanding cell health, as across these three replicates are three different passages that could actually change morphology over time. So we wanted to delve into this deeper. So using DeepCell, we were able to extract over 105 morphological features to help us improve cell characterization and actually start investigating some of these morphological features in more details. So DeepCell was able to extract statistical significant features such as image moments and ellipse elongation that indicate stress and structural asymmetry. So some of these features help us understand the morphological drift in control batches that can actually affect the yield, potency, and safety. So morphological variation, as we know, can skew comparisons with treated samples, and that these morphological drift in these control batches that we see can actually impact quite significantly, and passage differences can significantly impact yield that needs to be considered during biomanufacturing. And you can see here in this heat map, in a very detailed heat map, just how many candidate reference morphological features we can extract using DeepCell. So now that we can see that there's differences in control samples, what we wanted to also see is if DeepCell is able to determine cell line aging and actually identify various different clusters associated with cell line aging. So this is a major challenge in ATMP production. So cellular senescence or cell line aging, as I mentioned here, in biomanufacturing pose significant challenges to product quality, consistency, yield, and safety. And these safety concerns, such as tumorigenesis, have actually also been have emerged, which led to global standardization initiatives such as development of new ISO standards. So here, for example, the two new ISO standards currently in development include tumorigenicity assessment and also general consideration and requirements for cell viability analytical methods. Regulatory and ethical concerns, along with the need for standardization and clear guidelines, are also critical obstacles, and cellular heterogeneity, maintaining proliferative capacity and regenerative potential, preventing senescence and apoptosis is a challenge in clinical studies. So monitoring of morphological parameters during cell culture, for example, such as senescence and density loss, can be quite crucial in influencing the function of the cell product. So initially, the work that my team and I have done in the past is that we were able to use, so again, using the commercially derived cell line here, MRC5, that's commonly used for production of viral vaccines, was chemically treated to induce senescence, quiescence, and apoptosis. And these are some of the representative images that we've obtained using bright field images using our Tune site picks. And already they've been able to depict distinct morphologies. So simple morphological profiling reveals significant differences in cell diameter and eccentricity reflective of their cell health states. So of note, what we've also done here is we actually used fluorescence markers for us to check accuracy of gating strategies in bright field imaging and to actually isolate these populations. So what we wanted to do then is extend this line of investigation in DeepCell when we were able to use this as a demo to actually start investigating other influences and factors that would change and alter morphology over time during routine cell culture. So deep cell analysis showed that the healthy cells at low passage numbers, so I have distinct morphology compared to late passage cells. So for optimal function, MRC5 should be used between passage 1 to 20 upon thaw. And beyond this, they will start to show signs of aging and altered characteristics. So due to their finite lifespan of about 42 to 48 population doubling. Now, what we were able to show here is that Again, the healthy cells, passage 19, versus a late passage of passage 50, for example, and UV treated as a control, show significant area distribution within deep cells. So for the healthy samples, specifically clustering on one corner. What we were also able to show here is that these are these healthy passage P19, for example, is attributed to the true passage number of these cells and not upon thaw. So what we were able to show here is actually there's inherent differences within these healthy versus late passages, regardless of whether or not we were passaging this from thaw onwards. So what we want to look into further is using deep cell and its morphological clustering. We were able to reveal healthy cells, and they actually form distinct clusters in agreement with the previous slide. So whilst apoptotic and debris populations are more prevalent in late passage and UV treated cells. So as you can see here, the clusters 0, 2, 8, and 13 seems to demonstrate these intact cell populations along with the density and the distribution of these healthy in comparison to the late passage cells that you can see on this right-hand side here. Late passage cells tend to have clustering within the 3 to 7, 9 to 13, and 15 that are quite indicative of apoptotic cell fractions when we were looking at individual cells, like single cell images. Cluster 1 seems to show cell debris that's mostly found in UV treated samples as we've predicted, and cluster 5 and 10 are linked to later passages that are not found in healthy optimal passages. What we were also able to show here is that these clustering, according to deep cell analysis software, do show differences in terms of eccentricity, roundness, area, and also within the perimeter, further indicating that these clusters are specific subset of populations. So this clustering helps us understand population dynamics and also identify outliers within the populations. So again, using MRC5 as a healthy sample, we were able, as a baseline morphology, we were able to see these significant reductions in area, also within the serum solved and also over confluent samples. That could be indicative of active cell apoptosis and stress in both healthy, but also within the late passage samples. And there's also a significant difference in overall area and health versus in late passage, here contributing to changes in morphology as the cells age. So changes in morphological features such as parameter, for example, is found to be the most consistent between, significant between healthy and late passage cells. Further demonstrating that the late passage exhibits profound morphological changes that indicates altered cell phenotype and functionality, despite them being able to still grow and proliferate. And independent of this, what we wanted to also show was that we were able to see the same trends with deep cell within our previous analysis as well, using the attuned side picks with the reduction of the area that we see here in with MRC5 being suspension cells. Adherent cells that have become suspension. So finally, what I wanted to show you is the power of deep cell in terms of using the HFM model that they have and extracting a comparison of over 105 morphological features that demonstrated key morphological features that should be further examined. So for example, here when we were comparing healthy against late passage samples, ring with a feature that measures thickness of peripheral region suggests quantitative difference in terms of their cell appearance. In addition, image moment 2 and image focus, for example, also show significant differences, which may be attributed to cell enlargement, senescence associated morphology, increased heterogeneity, and accumulated DNA damage. So features such as positive fraction that seem to have decreased in overconfluent and serum stop samples can actually be used to provide a potential threshold for these senescent cells moving forward. So what we wanted to show you here is that we were able to start identifying these significant differences within these morphological features that would otherwise be missed in traditional gating strategies and simple morphological profiling. So to summarize, morphological profiling driven by DeepCell can be a critical tool for quality assurance in biomanufacturing and clinical research. We're able to analyze label-free and real-time analysis of cell shape, texture, and structure during routine cell culture in adherent cell lines, for example. This also ensures this enables us to detect senescence-associated morphological changes and tracks passage-dependent drift in cell populations that could identify early signs of these phenotypic instability. So over time, morphological profiling, for example, can be implemented during cell culture to develop quantitative thresholding of specific features. As I've mentioned, in addition to some commonly used metrics, such as area, perimeter, and centroid axis. So this will hopefully be able to facilitate standardized decision-making and custom gating strategies. So we can also use morphological profiling to flag outlier populations, enhance reproducibility in cell expansion, and quality assurance of the cell starting material. So this provides an innovative solution that can strengthen regulatory compliance through traceable image-based metrics. And current efforts are also being done to evaluate other significant cells, such as iPSCs, Hec-293s, and PPMCs. Thank you so much for listening to my talk. And as always, thank you so much for DeepCell, Megan and the rest of the team, as well as Tony, for helping us set up and analyze quite a lot of the samples. And if you would like to connect, please feel free to connect with me through LinkedIn. Thank you.
Awesome. Thank you so much. That was amazing. I really appreciate you sharing your work with the larger audience. I mean, understanding how to study cellular senescence and aging through the lens of morphology is pretty amazing. So I'm really, really glad that you were able to share that. We do have a couple questions. I know we are a little bit over time, but hopefully we can just take a couple minutes to go through some of the Q&A if you don't mind. I think this one is specific to Dr. Rivera's presentation. Someone asked, how do you see REMI impacting the overall study of cellular senescence outside of MRC5 use cases, and where do you see this going long term?
So what we were not able to show you because of timing is that we were actually able to see and use DeepCell in terms of identifying morphological features in, for example, diabetic and non-diabetic patient samples, as well as red blood cell PNH samples here. So what we're hoping to do with DeepCell in the future is that we start building these subsets and candidate images, basically, to be able to demonstrate what senescent cell looks like in these specific samples, and hopefully be able to use that as a candidate reference image for image analysis.
Awesome. Thank you so much. I see another question here. I think this one is more for Dr. Heckman's presentation. Someone asked, did you have any previous indication that morphology would be able to identify different profiles between healthy bone marrow samples and AML bone marrow samples before running on the REMI?
Yeah, so historically, AML is diagnosed in the old days by morphology, and this is just by standard histology. So bone marrow cells are smeared onto a slide, and a pathologist and then they're stained, and pathologists will look at cells. And so basically, yeah, we knew that there should be a difference in morphology, and this is why we thought REMI would be actually a nice fit in analyzing AML, because we should be able to see this difference, and it did. That was great.
Awesome. Thank you. I see another question. Someone asked, what is the most informative cell parameter regarding its morphology, and what is the most informative cell parameter regarding its morphology when you are trying to cultivate new primary cells? I think this can go to either presentation. Maybe for more primary cells, Caroline, do you have an inkling as to which either morphometric feature was more useful to you, whether you use the differential features tool to look at the morphometric features over the deep learning features.
Well, cell size is always a sort of a standard feature that we look at in terms of AML blast, but and then also we know that certain drugs will affect cell size. So cells or drugs that are cytostatic will cause cells to become a bit bigger, and then cells that might be impacting or inducing cell death will cause the cells to the membrane to start to disintegrate. So we should see some differences as well. But it was interesting because with the human foundation model, we mainly saw these deep learning features that were unexpected, but interesting and something that we look forward to following up with.
Great, thank you. We also have another one that says, would sample preparation processes impact the morphological features and introduce inconsistencies, particularly with frozen samples? I can also speak a little bit to this. That is a great question. Sample prep is very important when you're looking at understanding morphology and also standardizing morphology as a readout in these use cases. Um, the most important thing when you are preparing your samples is to remain consistent and optimize your processes to be gentle to the cells in order to allow them to stay in their most unperturbed state. So we have a lot of researchers that perform various methods of sample preparation, whether that's tissue dissociation, whether that's fixation, thawing from cryopreservation, pulling straight from cultured cells. It's just very important to remain consistent. I don't know if Caroline or Dr. Rivera or Jen, you want to speak to your sample preparation experience with REMI compared to other platforms, or how you felt about introducing inconsistencies to your data set.
Yeah, we've done quite a lot of biological replicates actually and technical replicates as we were testing deep cell. And what I can say to that is we didn't, because we were so used to flow-based assays where we had various default thresholding in place, what we did have to consider with using deep cell was that we see a lot more debris that may have otherwise been missed because we just have a higher threshold that you do. And because deep cell images every single one, even a little tiny debris, we had to be more strategic and be more careful in terms of our sample preparation. But what that did actually show us was that we did see some variation, specifically in diabetic and non-diabetic samples that we were able to run. And we saw differences in and how sample preparation influenced the morphological features. So we were actually able to see contamination from adipose tissue, so adipocytes that we didn't see before. So again, that was quite a good tool for us to do a quality control when doing our analysis in deep cell. That was useful.
Thank you. Yeah. Awesome. And Caroline, did you have anything to add?
Yeah, and I can just add that for our analysis, we were using viably cryopreserved cell samples, and as Megan already mentioned, it's important to be consistent. And I think one question we were sort of asking ourselves in the beginning, okay, do we do a fixation of cells after doing the thawing and trying to get rid of the debris? Do we fix the cells, or should we just analyze them straight? So we were thinking, actually, we wanted to do some follow-up by flow analysis. So we decided to go ahead and fix the cells. But it was really nice to see the consistency among the different healthy donor samples, but then also with the AML cells. So it's good to have also these replicates. Our replicates were mainly different disease samples and different healthy donor samples, but the results were all consistent.
Great, thank you. Yeah, and one more thing just to add on to that is, when considering what type of condition you want to run your samples in, it's important to consider, if you do want to do sorting, what your downstream applications are and what types of cell states are compatible with those assays. So if you want to do, some sort of functional assay, you have to consider whether fixed or live would be okay to run that. Or if you want to, do some sort of assay that requires live cells, obviously it's pretty interesting to be able to, do certain sorting schemes to maintain the viability of these cells. There's a lot of flexible workflows, so awesome. Okay, we are 10 minutes over, so I think we are going to end it there. Of course, if anybody has any other questions, feel free to reach out. I am Megan Burr. My email is megan.burr@deepcellbio.com, but there are a lot of ways to get in contact with us, whether it's LinkedIn, through our website. Feel free to reach out. Thank you so much for joining. And again, thank you so much to Dr. Rivera and Dr. Heckman for participating in this webinar. Your work is truly amazing. I'm inspired every day talking to you both. And I cannot wait to see where this goes. So thank you so much. Have such an amazing day. And yeah, have a great one. Thank you.
Thank you. Bye.
All right. Bye, everybody.